All right. I'm Warwick Bowen. It's my pleasure to chair the next session on quantum enhanced optical spectroscopy, and also to give the first talk, which is mainly an overview of the area and introducing the speakers. I wanted to start by thanking the organizers. I have so far really enjoyed this conference. It's great to see what I would describe as a new community coming together around applying quantum in biology and medicine. I think it will be a community that has major impact. I lead the Australian Research Council Centre of Excellence in Quantum Biotechnology, which really has the same sort of spirit as this community here. So we're a very multidisciplinary centre applying quantum into the life sciences, particularly applying quantum sensing, imaging, and computing into those areas. So if you want to come to Australia and work in that area or a national centre, you can choose between 5 different cities. Yes, all right. So I want to start with a very brief overview for those of you who don't know me of what we do in my lab. Basically, we do precision and quantum sensing and apply that into a range of different applications. One area which is probably not hugely overlapped with people here, but I find really fun, is superfluid physics. We study superfluid helium. This trace up here, turns out, is soliton fission, which is nonlinear dynamics that happens in shallow water wave physics when you have very thin films and very large amplitudes. It's been predicted in superfluid helium for 50 years, and we just observed it on the archive. So if you care about hydrodynamics and nonlinear hydrodynamics, then check it out. We also study quantum vortices and things like this in superfluid helium. We work on phononics, so this is learning how to control phonons on a chip and use them in applications. Just spun out a company called Sonic, who are applying this technology into hardware machine learning. And I think in the next few months, the whole research will go into the company, so I'll no longer be doing it myself, which is kind of exciting. And we do optomechanics, interest in quantum optomechanics, particularly how you can use continuous measurements to control and learn about mechanical systems and regimes where there is quantum back action. And classical optomechanics, applying optomechanics into precision sensing, particularly magnetic field sensing, ultrasound sensing, and inertial sensing. And what's becoming an increasingly large area in my lab is quantum bioscience, so applying quantum tools of various kinds into biology, and I'll tell you a bit about that today. Okay, so everyone here, I guess, knows what spectroscopy is. Essentially, it's taking a spectrum of something which gives you a fingerprint about what that thing might be. Generally, we would think about the spectrum being a frequency spectrum of the electromagnetic field. Could also be a mass spectrum, as we heard about from Amir today. What really makes spectroscopy powerful, at least with electromagnetic fields, is that you're not limited to just the visible range. There's an enormous range of frequencies that you can do spectroscopy over, and it turns out that spectroscopy across the whole range of frequencies that we are able to experimentally access is useful in biology. So let's run through some examples. So at fairly low frequencies, RF to microwave frequencies, you have MRI, you have NMR, you have EPR. You can detect chemicals, identify them, and image them, and build medical imaging devices. At higher frequencies, so terahertz frequencies, they're the frequencies that proteins collective modes act at, and at the frequencies of molecular bonds. So you can do Raman imaging, you can identify proteins, you can fingerprint them, and you can do imaging of the body and tissue. At higher frequencies again, at infrared frequencies, you're starting to get into the regime where the protein tertiary structure is detectable. And you can, if you've ever had your blood oxygen level measured with one of these little clips that gets put on your finger that's using infrared, visible and infrared frequencies together. And at visible frequencies, of course, we've got all of the powerful tools in light microscopy. So fluorescence microscopy we already heard about today, 2-photon microscopy, super resolution microscopy, etc. Higher again, you have X-rays and gamma rays, both of which are important for various applications of biology, from PET imaging, which is one of the most important medical imaging techniques we have, and actually involves quantum entangled photons, gamma ray photons, although you don't use the entanglement. And X-ray, of course, gives you protein structures. So spectroscopy is super, super important for biology and medicine. How can quantum help make spectroscopy better? I guess the first point I would make is that many, in fact, I would say most, of the spectral features we look for in biology arise from quantum mechanics. NMR is a very good example of that. But beyond this, there are various things we can do using quantum tools that make spectroscopy more powerful. One observation is that spectroscopy usually involves scattering light off some system, and a very small amount of the incident photons get scattered. It's a nonlinear process of the weak cross-section. That means you have faint signals, which you would like to make more visible. There are various ways that quantum can help you do that. One of them is if you use correlated pairs of photons, you can enhance 2-photon absorption for 2-photon fluorescence microscopy. You enhance the cross-section, make it linear scaling with intensity rather than quadratic. Another is you can use quantum correlated light to suppress the background noise of measurements of scattered photons. And in fact, enter regimes where you can measure scattering of less than 1 photon. And another area that we've heard a bit about today is you can bring new functional capabilities to spectroscopy using quantum. So if I put a molecule in the cavity, I change its density states, I change its behavior, and then I can learn about the physics of that molecule. Or if, as Peter was talking about earlier today, I can engineer clock transitions and things like this into a molecule, then I build new capabilities into it. So that's sort of, benefits of quantum itself, but I think there are other benefits which are equally or potentially even more important that our community can provide to the biology and medicine community in spectroscopy. Those benefits come from the fact we try really, really hard to push noise down and make really good clocks and make really good systems. So I'm giving you 3 examples here. The first one is using entanglement between a visible or near-infrared photon and a terahertz or mid-infrared photon to do terahertz or mid-infrared imaging without any terahertz or mid-infrared components. So that means that you don't get better performance out of your system, but you can make the whole system much cheaper than you could if you relied on something like a terahertz camera. So while we think about quantum as being complex, in some situations you can actually reduce complexity and cost by employing quantum physics. The second we already heard about a little bit today, frequency combs. Frequency combs are a very powerful tool that came out of our community and is very useful for spectroscopy. And the third here people will recognize is one of the LIGO interferometers. LIGO spent, and a whole community around LIGO, spent decades figuring out how to get rid of classical noise sources so that they could operate interferometers at quantum noise limited performance. Those sorts of techniques can be directly applied into biology to improve biological measurements. Okay, cool. That was my overview of quantum optical spectroscopy. Now I want to spend a little bit of time talking about a few examples from my lab at different frequencies. The first one of those is quantum control of protein vibrations. The idea here is that it is believed that it's known that the motions of proteins are critical to their function. We need to go beyond basic theories of molecular interactions in biology, which are these lock and key mechanisms where two things have complementary shapes and bind, to a model which models reality better, because the reality is that proteins are always moving around and it's only at a particular time that they might be able to interact. So it's known that motions are really key to understanding protein function. We don't have good tools to measure those motions at the speeds at which they occur. And we don't have good tools to diagnose the effect of motions, or at least our tools are limited when it comes to diagnosing the effect of motions on actual function. For example, a standard method that people will use is that if you've got a protein of a certain kind and you think a certain motion influences its function, maybe drives a conformational change or some enzymatic reaction, then you'll genetically engineer the protein so it moves in a different way, and you'll see if that affects its function. But of course, when you genetically engineer something, you change lots of things. So what we are trying to do in my lab is apply quantum control techniques, laser cooling techniques from atom optics into vibrations of proteins. So instead of genetically engineering them, I can switch particular vibrations on and off and look at the function of the protein. We haven't got there yet, but we've made some good progress, I think. This is what a typical device would look like. This is a 1D metamaterial cavity. That's the cavity in there. It's on a fiber and it's dunked into this sample tube. And the idea is that a protein, we design these devices, they're similar to the ones that Jen talked about earlier today, but designed for protein interaction. So we design them so that proteins can be trapped optically inside this cavity, and then we can use the cavity enhancement to measure the protein dynamics very quickly, and ultimately to do laser cooling or control of the vibrations of the protein. This is an example of a measurement. This is ferritin. I think Amir had some ferritin in his slide too. It seems like it's the slide, so it seems like it's the protein to study. It's very big, which is helpful, and it's spherical, which is kind of nice. We also have similar data for much smaller proteins down to about 30 kilodaltons. But here I'm showing you that we can trap ferritin for extended periods of time. The noise here is larger than the noise here. That tells you that we're measuring the motions of the protein. And if I take the Fourier transform, I find that I can measure those motions up to about 10 megahertz frequencies, which is about 3 orders of magnitude faster than other techniques. And that comes about from the cavity enhancement. Okay, that's example number one. Example number two is quantum Raman imaging. We already heard about Raman from Jen, maybe other people this morning. It's a very powerful technique. The idea is you can get a spectrum of the concentration of different chemicals in a sample, widely applied in biology. It turns out that you can tell the difference between cancerous and healthy cells, for example. You can even measure action potentials in neurons from their Raman shifts, many other applications. The problem with Raman, as I mentioned earlier in general, is that the interaction with light, the Raman interaction is pretty weak. So what you find is that precision Raman microscopes are always working at this bleeding edge, where they're using as much power as they possibly can without perturbing the sample too much to make them fast. And by fast, I mean imaging in minutes typically, sometimes faster than that, depends on the Raman bond that you're imaging. So they're slow, but they're powerful. And it turns out the best Raman microscopes now are shot noise limited. And that means the only way you can make them better is to use quantum effects. And that's what we demonstrated a few years ago now. So this is a Raman image of a yeast cell using a standard high-performance Raman microscope. If we apply squeezing, we can we can get an image which has a higher contrast. And now, after some years of work, we've improved the speed of imaging so that we can image this cell in about 15 seconds, and we're able to track the dynamics of lipids moving around inside the cell. Okay. The third example I wanted to give, I don't know how I'm doing for time. I feel like I'm going fast. Maybe I am going a bit fast. Okay, good. The third example I wanted to give is an example of quantum limited imaging. So we're trying to apply those techniques that are used in gravitational wave detection without using any quantum resources. And here is the problem we're trying to solve. It's a really basic problem. If I give you a cell and I ask you, is the cell alive? Is it dead? What phase of its life cycle is it in? Is it happy? What's it doing? Right? It's actually surprisingly difficult to answer all of those questions. If I put that cell under a standard phase contrast microscope or something like this, then I just see structure. I can watch for a while. If it divides, I knew it was alive, right? But inside that cell, all the time, there's nanoscale activity going on that's keeping it alive, right? This is what this image is supposed to show. This is the cytoskeleton, all these motor molecules in the cytoskeleton that are driving it around. So you expect that there should be signals in there, even though you can't see them. Now, the normal way you would answer the question, is it alive or what is it doing, is you would genetically modify it to express a certain fluorescence if it's in a certain state, or you'd label it to emit light if it's in a specific state. But in many cases, you don't want to do that. Bacterial antibiotic resistance is a very good example. We can take a sample out of someone. You can't genetically modify the bacteria because they're natural bacteria, right? So the question is, can you see this activity inside the cell? And the answer is you can. There are techniques that do this. They typically use optical tweezers. We heard about particle tracking also earlier today, which was nice. This is a cell with a particle in it. If you watch that particle move around in a cell, you don't see Brownian motion. You see something else because the cell is active. There are forces that are driving the motion of that bead, and also it's a congested environment, so it just can't move freely. And that tells you about the material properties of the cell and also its activity. The problem here is that A, I have to put that bead in the cell, which is not ideal. And B, I only get a point at that I only get data at that one point where the particle is, and I don't necessarily have the ability to control that point. So we developed a new technology, which I think resolves these challenges. It's based on a type of microscopy called iSCAT microscopy. Here is it turns out it's a very simple microscope, and basically you measure phase differences between scattered light in my sample. So there might be a cell here, and I've got some proteins in the cell that are scattering light, and they interfere with light. That interferes with light that reflects off the cover slip. And the idea is because the path lengths are almost identical, you have very little phase noise between those two fields, and you can get a very quiet measurement. We couple that with a very low noise illumination laser source. And when we do that, it turns out that if I measure light on this detector here, this is the shot noise limit. I see all of this activity inside the cell that's due to the motor molecules driving things around. And that's a spectrum, tying back to the topic of the presentation. It turns out I can fit to that spectrum, and I can learn a lot about what's going on in the cell. It turns out, and this fit is for a model that's to describe the activity of the cytoskeleton of a cell. It turns out there's some thermal activity. Maybe I shouldn't call that activity. That would be there if the cell was dead. There's some active motion, and there's some active motion, and I can extract 4 parameters. I'll give the game away too much here. I can extract 4 parameters about the sample. I can extract the temperature of the active dynamics. and I can extract some microbiological parameters, which I won't tell you about today because I don't have time. I'm going to give you an example. This is a epithelial cell. It's a cancer. It's a cancerous epithelial cell, A549s. This is a standard phase contrast image of the cell. This is an example of this temperature image. It's getting all the same information as in the optical tweezer example I gave, but now across the whole cell, imaged with no probe particles. And you can see this is the active temperature. It's about 10 times 300 Kelvin. The cell's alive, right? But what's cool is, in this example, if we apply some treatment which the cell doesn't like, this PFA cross-links all of the proteins in the cell and causes it to die, then after some time, we can see the activity in the cell changing. So here, this part is now at 300 Kelvin, it's dead. And this part, the cell is going, oh shit, I'm dying, I better do something. So it's more active. And we can see all this activity now that was previously invisible. Yeah, so that was my 3 examples. Now I get on to probably the more fun stuff for you guys, the 3 other talks from the session. We're very fortunate to have 3 great speakers, Marcus Ant from University of Vienna, Naisin Quan from Colombia, and Huzong Liang from Colorado. Marcus of course, is well known for his work on double slit type interference of molecules. He'll tell us about that, but I think also about some biological applications of that sort of technology and some other work that he is doing on molecular beams and trapping molecules, mass spectroscopy perhaps. Naikin is working on stimulated Raman microscopy, so similar to the work that I showed you from my lab using squeezed light, but in her case, applications in chemical imaging of cells and things like this. And Kui Zhong, apologies for pronunciation, works on cavity enhanced frequency cone spectroscopy at Colorado, and will tell us about applications of that for breath testing. With that, I will conclude, and I'll just say 2 things. One is remind you again, if you want to come to Australia, get in touch with me. Would love to talk to you. And the other is that we have a whole lot of other work that I didn't have time to tell you about going on. I'm giving you 4 examples here. Understanding transmembrane drug delivery, using magnetic brain imaging for concussion diagnostics, taking it out into sports fields, deep tissue imaging using quantum correlations, and super resolution imaging using structured detection. So if you're interested in any of those things, just come and have a chat to me. Thanks.
To ask you questions.
All right, I'm in charge. Yes.
You just teased us with these things.
I did, yeah.
How do you define a local temperature? Can you clarify what you're actually doing there? I was really interested in that.
I can attempt to clarify that. So, so there are models of how particles diffuse in the cytoskeleton of cells. If you include forces from the motor molecules that are the reason that your cells have structure is that there are microtubules and motor molecules move along those microtubules. Those motor molecules, you can model the forces from them. They're like an impulse force, like a random telegraph noise that has a different spectrum to thermal noise. And so if you work out the susceptibility of the local cell to a given force, and you know the thermal force spectrum and the active force spectrum, then you end up getting a total spectrum that you can fit to extract the temperature of the active forces. I don't know, how did I do?
No, that was great. Thank you. Thank you.
That's actually pretty new stuff for me. We only figured out we could do that model about 2 weeks ago. So Yeah.
So it's just curious about this iSCAD thing. Typically, that's good enough for seeing single proteins, even to small masses. But there's a lot of stuff in the cell. Huh? So there must be a huge signal, kind of. So how fast is this, and how much can you tell about what you really see inside?
That's a very, very good question, and my answer is going to be a little bit speculative, but I'm fairly confident I understand now that it puzzled us for a long time why we saw anything, and why we started making measurements, and the measurements perfectly agreed with the spectrum you get from a single point measurement with a probe particle, right? So then you think, why should this complicated measurement of all sorts of dynamics going on in the cell exactly mimic what I get? We did measurements with a particle as well, and they're the same. How on earth is that possible when there's so much, and it turns out two things are important, I believe. One, so we'd initially thought that we were seeing scattering from mitochondria because they're like the biggest common object inside cells. But it turns out that if you have correlated motions, then the signal you get is amplified by the number of particles which are moving in a correlated way. And it also turns out that it's known that particles in cells, you have the cytoskeleton that's sort of moving and everything gets sort of drawn along with it, right? So there's a lot of correlated motions in cells. So what we believe is going on is inside the voxel that we're probing, you have proteins, single protein molecules, many of them moving in a correlated way, and that's what we're detecting. So we're precisely detecting the motion of the cytoskeleton through this correlated dynamics of proteins. And you do the numbers, and it seems like that's right, but it's a very hard thing to prove. I'm not actually sure quite how to prove it. But that's the belief, is we're just directly measuring this sort of swaying of all the proteins in the cytoskeleton due to the cytoskeleton. Yeah. Should watch the time. I'm in charge here, right? I've probably run out of time. So maybe we will thank the speaker and